Now let's study about dominant epistasis. You know what is meant by epistasis, you know what is meant by dominant epistasis also. In epistasis, there are two genes which are located in two different loci that control a particular character, a single character. Okay. Here, the particular or the peculiarity is that one gene overmasks the expression of the other gene. That is epistasis. You know what's meant by epistatic gene and hypostatic gene and all. Anyway, if the dominant allele or alleles of one gene overmask the expression of the other gene, then we call it as dominant epistasis. So, once again, in dominant epistasis, the epistatic gene is the dominant allele or alleles of one gene. Okay? So, here the dominant allele of or the dominant alleles of one gene overmasks or suppresses the expression of the second gene. Second gene may be either in the dominant condition or in the recessive condition, right. So, let us study with the help of an example. You know cucurbita, pepo, that is summer squashes. There are three fruit colors are there: yellow, white and green. Here the yellow fruit color is due to the presence of a dominant allele of Y gene, that is capital Y is responsible for yellow fruit color, and capital W, that is the dominant allele of W gene, is responsible for white fruit color. And when the alleles, dominant alleles of both the genes are absent, it leads to the formation of green fruit color. What may be the genotype? Double recessive condition, that is small w, small y condition. So, in other words, we can say the fruit color of summer squashes or cucurbita, pepo is controlled by two different genes, say W and Y gene. Okay. So, here the very important thing you have to remember is, even if capital Y is present, whenever capital W is present along with, the expression of Y, dominant Y allele is suppressed. That is, I told you that capital Y gives rise to yellow fruit color, but that yellow fruit color is never produced if the capital W allele or the dominant allele of W is present along with. So, W is the dominant epistatic gene. Okay. Now, let us see what will be the F1 individual which is produced from a pure white parent by crossing a pure white parent and a green parent, and let us see what are the different gametes which are produced from the F1 individual, and also the F2 phenotypic ratio and the different genotypes of offspring which are produced by selfing of F1 individuals. Okay. So, this is regarding the dominant epistasis, and we are studying with the help of cucurbita, pepo, that is summer squashes. This is the genotype of white pure parent, capital W, capital W, capital Y, and this is a genotype of pure green parent, small w, small w, small y, small y. Why is it white in color? This capital Y, the expression of capital Y is inhibited by capital W or dominant allele of W gene. So, this is the dominant epistatic gene and this is the hypostatic gene. Okay. So, this is the only possible gamete which can be produced from this parent, and this is the possible gametes that can be produced from this parent. All these things you know very well. Now, coming to the F1 organism, this is the genotype of F1 organism, capital W, small w, capital Y, small y. And what is the phenotype? So, though capital Y is present, capital W is also present. So, capital W overmasks the expression of capital Y, and so it is white in color. So, white fruited plant is the F1 phenotype. Now, what are all the different gametes that can be produced from this? Capital W, capital Y, here capital W, small w, capital Y, and small w, small y. So, these are all the different 4 types of gametes which can be produced from this F1 individual. Now, what about the F2? So, upon selfing the F1, we get F2, right? Now, let us see the selfing of F1 organisms. Okay. So, these are the gametes and these are the gametes formed from F1. So upon selfing, you can see the different offspring which are produced in the F2 generation, and all these are shown in this Punnett square. So this is first organism is capital W, capital W, capital Y, capital Y. Of course, it's white in color because capital W is present, though capital Y is also present because this is the dominant epistatic gene. You know now that. Okay. So here also capital W, capital W, capital Y, small y. So capital W is present. So you don't have to think about anything else. It's white in color. Here also white. Here also white. Here also white. White, white, white. Here, here. But now here you can see only small w is present. That is, the dominant epistatic gene is absent and dominant alleles of Y gene is present. So it is yellow in color. Similar situation here also. Here also you can see yellow fruit color because capital W is absent or only the recessive allele is present there. Okay. Then in these cases also it is white in color because capital W is present. Right? And this is the last case, that is a green phenotype. Here dominant alleles are completely absent. It is double recessive organism. Okay, it is green in color. So when we count the number of organisms, S2 phenotypic ratio becomes 12:3:1. That is 12 white, 3 yellow and 1 green. So the F2 phenotypic ratio of dominant epistasis is 12:3:1. Okay.